by Ryan Zenhausern, and he's going to talk about developing lipid nanoparticles for non-liver RNA delivery of gene therapy. Thank you.
Hello, everybody. It's very nice to be here today. I'll be presenting on behalf of my PI, Dr. James Dahlman, who unfortunately couldn't make it, but I'm happy to be with you all for my first meeting. So RNA therapies, as we all kind of know, are exciting new ways to treat disease that work to either silence, replace, or edit the genes that cause disease. And so using RNA as a therapeutic molecule can be difficult in that these molecules can easily degrade. Check this up. Is that better? A little better? So using RNA as a therapeutic molecule can be difficult in that this can degrade easily and can't easily pass through cell membranes. So we need delivery vehicles to bring them to the cell types where they can exert their function. And lipid nanoparticles represent one such delivery vehicle that have been used in previously FDA-approved drugs. So in 2018, we saw the approval of LNP carrying an siRNA for the treatment of a liver disease. In 2021, saw 2 approvals for the intramuscularly administered LNP mRNA COVID-19 vaccines. So through these approvals, we've seen that LNPs are safe, scalable, and effective delivery vehicles. And there's been a huge interest in using these vehicles in delivering RNA therapies for treating other diseases. The challenge with using LNPs, though, is that after systemic administration, most of these LNPs end up in the liver. And this has been very helpful if you're trying to create treatments for liver diseases. We've seen great data in that kind of regard. But ultimately, if we want to create therapies for other organs, we ultimately need to identify LNPs with non-liver tropism. And so a typical lipid nanoparticle is composed of 4 components, including an ionizable lipid, a cholesterol component, a pegylated lipid, and a phospholipid. By changing the individual chemical identity of any one of these components, as well as the relative molar ratio between components, we can shift where these LNPs go in vivo. Oh, yeah, of course. Great. So the challenge with developing new LNPs is that there are many possible chemical spaces for any one of those components, and there are many possible combinations in which we can mix them together to make LNPs, right? And so the chemical space that we need to survey here is very large. So our lab has used DNA barcoding as one way to kind of quickly scan these diverse chemical spaces. And so the way that this essentially works is that we incorporate a unique DNA barcode into our unique lipid nanoparticle formulation, such that we can create libraries of these unique LNPs, each with their own unique DNA barcodes and a shared kind of common reporter mRNA. We can then pull these libraries together and then inject them in the mice. We can then look at cells in a variety of different tissues across the mouse to identify where there was functional delivery, so where there is reporter mRNA turned into a reporter protein, and then profile the relative abundance and the presence of different barcodes to essentially backtrack what particles went where and where do they work. And so there's a few kind of systems that I'll talk about as I go through this talk. But essentially, we use these barcodes and their reporter mRNAs all in unique LNPs to create these high-throughput LNP screens. So I am also not a CMT researcher, but I am going to give you an example of how we can use our screening technologies to identify non-liver LNPs and then kind of tie that in towards the end of the talk. And so I'm going to talk about some of our more recent work in developing lipid nanoparticles targeting hematopoietic stem cells, or HSCs, kind of as an example of how we use some of our more recent screening systems. So HSCs are a therapeutically relevant cell type as well, given that the disease of this single cell type can give rise to a variety of blood disorders. So theoretically, by correcting the disease of this one cell, you can lead to treatments in a variety of different blood disorders. But ultimately, this requires RNA delivery to those hematopoietic stem cells directly in vivo. And this is challenging kind of for 2 reasons. The first, that we need to identify LNPs that are able to escape the liver. And second, we actually need LNPs that can go and functionally deliver to the cell types of interest that we're trying to hit. And so although this cell type has been really exciting and people are trying to target these cells, there are very few examples still of LNPs that escape the liver and target HSCs. And this is in part because these cells are fairly rare. and they represent about 0.01% of nucleated cells in the bone marrow. So to kind of address this problem, we applied one of our more recent screening technologies that combines DNA barcoding and single-cell RNA sequencing, such that we can then perform delivery experiments in individual transcriptomically defined cells. So here's an example of how we went through this workflow for screening these non-liver LNPs. So first, we created a unique library of LNPs, each with their unique DNA barcodes. We created about 120 of these LNPs. We then pooled them and then injected them into the mice. From the same mice, we then isolated bone marrow cells and enriched for the fraction of cells that we're interested in looking at, which is this lineage negative fraction, and performed our single-cell multiomics approach on this. And then from the same exact mice, we also profiled delivery in the key off target of the liver, kind of at the cell type resolution using flow to identify delivery to the liver. So in general, just using both of those data sets, we're able to quantify how every single particle in our screen was able to deliver RNA to our on target of those HSCs, as well as to our off target of the liver. And we're able to rank all 120 LNPs based on those ratios. So using that information, we're able to identify 3 lead candidates from this screen, which we then wanted to individually confirm. So we then individually formulate those LNPs and then inject them in vivo. And we can then isolate tissues from around the mouse to identify the distribution of these LNPs across a variety of cell types throughout the mouse. And we do this using flow cytometry. And so I'm going to show you a few examples or I'll show you the 3 lead candidates we identified. And what you're going to see are these radar plots, which are essentially, show the amount of delivery in the different cell types across the different organs as different points around the circle. And so here's what it looks like when we have our untreated group. Here's what it looks like when we have we add a liver-specific LNP. And then here's what it looks like when we have our 3 lead candidates. So essentially what we're seeing here is that we're able to target bone marrow cells with one of our formulations, and we're able to see an on to off target ratio that approximates 1-to-1 between in our bone marrow cells and our liver. So we performed these experiments all in mice, but we also wanted to see how these delivery profiles stacked up in non-human primates. So in collaboration with UC Davis's Primate Center, we formulated these LNPs and then injected them into rhesus macaques, and then isolated cells from the bone marrow, specifically those CD34 positive fractions that are enriched for those stem cells we're trying to hit, as well as cells from the liver. And we looked at the delivery across a range of different doses. And so here are the results from that experiment. And ultimately, what we're able to see are kind of 3 main things. The first, that we're able to get delivery to the CD34 positive fraction in these NHPs. The second is that we're getting that on to off target ratio between our CD34 positive fraction and our liver cells that approximates that 1-to-1, which is similar to what we had seen in the mice. And then finally, we were able to see delivery at our lowest dose of about 0.25 milligrams per kilogram. So I think in sum, I wanted to use that as an example for how we can use these high throughput LNP screening systems to identify non-liver LNPs and how we go about characterizing those delivery profiles, both in mice and ultimately in non-human primates. I also wanted to kind of change tacks a little bit here and describe some of our work that we had done in investigating RNA delivery to the brain. So like I mentioned before, there's kind of 2 main things that we're interested in. The first is being able to escape the liver, and the second is being able to actually target the cell types we're interested in hitting. So another grad student in our lab, Yanina, was been in charge of this project and has been exploring RNA delivery from previously characterized non-liver LNPs to the brain. So the idea here being, if we have a known LNP that actively detargets the liver, is it able to free up more nanomaterial to go to other places in the body? And so what she did was to formulate and inject this LNP, which has very low liver delivery, and is in fact actually a lung vehicle, and take a variety of different organs that weren't taken in the first pass of the experiment. And what she found was that there was a measurable but low amount of functional delivery to the brain when measured at kind of a tissue level. So she wanted to go through and explore this using flow cytometry to explore, what cell types she was actually hitting more specifically. And what she actually ended up finding was that digesting these organs has been really difficult, has led to low sample quality, and led to variable readouts of this already low level of delivery. So she faced a challenge there just in confirming this lead particle, which also kind of hindered her efforts in performing these high throughput screens. So the main lesson from that was that we needed to find a way to characterize this delivery directly in situ without the need to kind of digest these organs. In this case, the brain, but in other cases, any other organ that may be fragile or difficult to digest. And so here she was able to kind of run the same experiment, but in this case, to characterize the delivery using a spatial RNA sequencing based platform, which is 10x's Xenium platform. And so here, what she was able to see is that you can identify a variety of different cell types based on their RNA transcripts in these tissue slices, and then overlay that information with her delivery information. And so what we were able to ultimately find is that we could see the different cell types we were actually delivering to in their spatial context. And so I guess the main lesson from what we've learned in that approach was that we're able to actually quantify the delivery when looking at it in kind of different ways when preserving that tissue's native kind of structure. So what we're going to be doing kind of going forward is incorporating the spatial RNA sequencing based techniques with our DNA barcoding, such that we can perform kind of spatial LNP screens. And we want to use that information as well as kind of new LNP chemistries in order to identify LNPs that might have improved non-liver delivery, in particular targeting other organs like the brain and nerves. That was a little fast, but thank you everybody for listening. I appreciate it.